This is Edward Bernetti with Grand Canyon University Anatomy and Physiology 202 lecture and my presentation on chemoreceptor control of breathing. What are chemoreceptors? A chemoreceptor is also known as a chemosensor. I may use those interchangeably. They detect certain chemical stimuli in the environment. In the way of controlling breathing, chemoreceptors detect levels of carbon dioxide in the blood. And to do this, they monitor concentrations of hydrogen ions in the blood, which decrease the pH of the blood. This is a direct consequence to the increase of carbon dioxide concentration due to carbon dioxide becoming carbonic acid in an aqueous environment. There are two main respiratory centers in the brain. We'll be touching on both of them, one being the medulla oblongata, which is the lower half of the brainstem that contains cardiac, respiratory, vomiting, and vasomotor centers and deals with autonomic involuntary functions such as breathing, heart rate, and blood pressure. The medulla sends nervous impulse to the external intercostal muscles and the diaphragm via intercostal nerve and phrenic nerve respectively to increase breathing rate and the volume during inhalation. The second respiratory center I'm going to speak about is the pons. The pons is located superiorly to the medulla and inferior to the midbrain. It is also ventral to the cerebellum. It typically measures about 2.5 centimeters in an adult, which is about an inch. The pons contains the pneumotaxic center, which regulates change from inhalation and exhalation, and the apneistic center, which coordinates the speed of inhalation and exhalation. Inside the pons, the first being the apneistic center, which sends signals to the dorsal respiratory center in the medulla to delay the off switch signal of the inspiratory ramp provided by the pneumotaxic center of the pons. It controls the intensity of breathing. The apneistic center is inhibited by the pulmonary stretch receptors. The pneumotaxic center is located in the upper pons, limits the size of tidal volume, and secondarily, it regulates the respiratory rate. A normal breathing rhythm persists in an absence of this center. It is also considered the antagonist to the apneistic center, which produces abnormal inspiratory gasps. We're going to touch on some nerves of respiratory control, one being the phrenic nerve, which originates in the neck between C3 and C5, passes down between the lung and the heart to get to the diaphragm. It passes motor information to the diaphragm, also receives sensory information from it. The next two nerves are cranial nerves, one being the vagus nerve, which is, as we all know, cranial nerve 10. It originates between the pons and the base of the stem of the brain and the spinal column. And the way this helps us in the respiratory system is that the lungs contain stretch receptors. When the lungs expand during inspiration, the stretch receptors of the lung walls are activated and act via the vagus nerve to inhibit the respiration center in the medulla and allow the reflex of expiration to occur so we don't over-inhale. The second cranial nerve we're going to talk about is the glossopharyngeal nerve, and it's cranial nerve 9. It exits from the brainstem from the sides of the upper medulla, and this is a peripheral chemoreceptor, which we'll touch on to in a minute. Central chemoreceptors first, which is one of the two types of chemoreceptors. They're located on the ventral surface of the medulla. They're located near the point of exit for the glossopharyngeal and vagus nerves, and only a short distance from the DRG. Central chemoreceptors communicate directly with the inspiratory center and are most important for minute-to-minute control of breathing. These receptors are very sensitive to changes in pH of cerebrospinal fluid. Decreases in pH of cerebrospinal fluid produces increase in breathing rate or hyperventilation, and it increases the pH of the cerebrospinal fluid, which then decreases the breathing rate to hypoventilation. Medullary chemoreceptors respond directly to changes in the pH of CSF and indirectly to changes of arterial PCO2. In the blood, combines reversibly with H2O to form a hydrogen ion and HCO3 by familiar reactions. Is relatively impermeable to hydrogen ions and HCO3. These ions are trapped in the vascular compartments and do not enter the brain. CO2, however, is quite permeable across the blood-brain barrier and enters the extracellular fluid of the brain. CO2 is also permeable across the brain and CSF barrier and will enter CSF fluid. In CSF, CO2 is converted to hydrogen ions and HCO3, which increases arterial PCO2, which in turn creates an increase of PCO2 in the cerebrospinal fluid and an increase of hydrogen ions as well to decrease the pH. Central chemoreceptors are in close proximity to cerebrospinal fluid and detect an increase in pH or a decrease in pH, and then send signals to increase or decrease breathing. Central chemoreceptors eventually do desensitize. Are peripheral chemoreceptors. They are receptors for O2, CO2, and hydrogen ions. They are located in the carotid bodies at the bifurcation of the common carotid arteries and in the aortic bodies above and below the aortic arch. Information about arterial PO2, PCO2, and pH is relayed to the DRG via the glossopharyngeal and vagus nerves, which orchestrates the appropriate changes in breathing. And these types of chemoreceptors do not desensitize. In arterial blood composition is detected by peripheral chemoreceptors and produces an increase in breathing rate. Decreases in arterial PO2, which is the most important responsibility of these receptors to detect the change of arterial PO2. Once arterial PO2 has decreased to less than 60 millimeters of mercury, breathing rate becomes very increased, steep, and linear. The next change is an increase in arterial PCO2. As the peripheral chemoreceptors can detect this, they're much less effective, and it's not as important to them as it would be to the central chemoreceptors. They also detect decreases in arterial pH. Decreases in our pH can cause an increase in ventilation, mediated by peripheral chemoreceptors for hydrogen ions. This effect is independent of changes in arterial PCO2 and is mediated only by chemoreceptors in the carotid arteries. Metabolic acidosis is a decrease in arterial pH. The peripheral chemoreceptors are stimulated directly to increase the ventilation rate in order to attempt to bring the pH back up to a less acidic state. The blood-brain barrier is a highly selective, permeable barrier that separates the circulating blood from the brain extracellular fluid. in the central nervous system. The blood-brain barrier is formed by capillary endothelial cells, which are connected by tight junctions with an extremely high electrical resistivity. This allows the passage of water and some gases and lipid-soluble molecules by passive diffusion, as well as selective transport of molecules such as glucose and amino acids that are crucial for neural function. CO2 can cross the blood-brain barrier as it is smaller than oxygen. The dorsal respiratory group, otherwise known as the DRG, is involved in the generation of respiratory rhythm and is primarily responsible for the generation of inspiration. It is stimulated via the apneistic center in the lower pons and is also part of the solitary tract, which is responsible for appropriating responses to the sensory information from the chemoreceptors and mechanoreceptors. It is inhibited by the pneumotaxic center. And the other respiratory group, being the ventral respiratory group, or the VRG, contains both inspiratory and expiratory neurons. The VRG is secondarily responsible for initiation of inspiration activity. After the dorsal inspiratory group, the VRG is responsible for motor control of inspiratory and expiratory muscles during exercise. Muscles of ventilation. I'm going to touch on a couple in detail and most of them very briefly. The first and probably most responsible would be the diaphragm, which is responsible for 45% of the air that enters the lung during quiet breathing. During inhalation, the diaphragm contracts, which enlarges the thoracic cavity and reduces the intrathoracic pressure, or simply saying the enlargement of the cavity creates a suction to draw air into the lungs. When the diaphragm relaxes, the air is exhaled by elastic recoil of the lungs. Internal intercostal muscles. There are several groups of muscles that run between the ribs and help form and move the chest wall. The intercostal muscles are mainly involved in the mechanical aspect of breathing in quiet and forced inhalation. They originate on the ribs 1 through 11 and have their insertion on ribs 2 through 12. The external intercostals are responsible for the elevation of the ribs and bending them to more of an open position. The internal intercostal muscles are muscles that aid in forced expiration. They originate on ribs 2 through 12 and have their insertions on ribs 1 through 11. The internal intercostals are responsible for the depression of the ribs and bending them inward, thus decreasing the transverse dimension of the thoracic cavity. Muscles of ventilation would be as follows. For inspiratory, the sternocleidomastoid, anterior serratus, scalene, serratus posterior superior, levator costarum, and for expiratory would be the internal intercostals, rectus abdominis, external and internal obliques, transverse abdominis, serratus posterior inferior, transversus thoracis, and parietal. Under resting conditions, expiration is passive and associated with the recoil of the lungs. Of breathing I'm going to cover on the next two slides, one being hypercapnia, which occurs when there's too much carbon dioxide and waste products from respiration in the blood. This typically is accompanied with respiratory acidosis, normally a trigger reflex which would increase breathing and access to oxygen, such as arousal and turning your head during sleep. A failure of this reflex can be fatal, such as sudden infant death syndrome. Hyperventilation. This occurs when the rate and quantity of alveolar ventilation of carbon dioxide exceeds the body's production. Hyperventilation can be voluntary or involuntary. Hypoventilation occurs when ventilation is adequate to perform needed gas exchange. It causes an increased concentration of carbon dioxide, or hypercapnia, and respiratory acidosis. Other patterns of breathing include eupnea, which is normal breathing, hyperpnea, which is an increase in pulmonary ventilation matching metabolic demand, orthopnea, tachypnea, which is an increased frequency of respiration rate, apnea, which is an absence of breathing, and dyspnea, which is difficult or labored breathing. I'm going to put out there is blood pH. You've heard me talk about this throughout the entire presentation. So arterial blood has a pH of typically 7.41, and venous blood being a little more acidic at 7.37. And when exercising, venous blood can drop down to a pH of about 6.9. That's it. Thank you for watching my presentation, and hopefully you feel a little more educated.